Start tank bioreactors have been designed for option A, availability of oxygen throughout the process. Start tank bioreactors are designed with an agitation system, that is with stirrer or impellers, to ensure uniform mixing and distribution of oxygen throughout the culture system. And this is very important for the growth and metabolism of aerobic microorganisms during the fermentation process. Hence, it is the correct option. Option B, ensuring anaerobic conditions in the culture vessels. This is incorrect because these type of bioreactors are mainly designed to facilitate oxygen transfer for aerobic process. And anaerobic conditions, if required, can be achieved by eliminating oxygen and not through stirring. Option C, purification of product. It is also incorrect because these bioreactors are used for cultivation of cells or microorganisms and not for downstream process of purification. Option D, addition of preservatives to the product. It is also incorrect because preservatives, if needed, can be added during the production formulation stage, but not during the culture or the fermentation process, and that is carried on in this start tank bioreactor.